Hello, everyone, and welcome to today's webinar, Incorporation and Automation of RNAscope Technology into Your COVID-19 Viral Pathogenesis Research. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems and Bio-Techne. To learn more about our sponsors, please visit Leica Biosystems and Bio-Techne.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Jyoti Sheldon, Andrea De Biase, Damian Cockfield. For complete biographies on our speakers, please visit the Speaker Biographies tab at the top of your screen. Jyoti, you may now begin your presentation.
Thank you so much for joining the webinar today. My name is Jyoti Sheldon. I work as an application scientist at Advanced Cell Diagnostics. And I'm going to go over today what are the different novel applications of RNAscope and how you can apply the RNAscope assay in your COVID-19 research. So we're going to learn about incorporating this highly sensitive and specific technology for your COVID-19 viral pathogenesis research. On the agenda today, I'm going to cover the technology itself. I'm going to go over our COVID-19 product offerings. And then I'd like to show you some examples of the publications that have come out showing how researchers have used this technology in their COVID-19 research. So to begin with, what is the RNAscope in situ hybridization technology? The RNAscope assay is a highly specific and sensitive method, and that is the value prop of the assay. This assay is a method that allows you to detect RNA biomarkers in cells and in tissues while really giving you that morphological context, that spatial analysis, something that you miss with a lot of other traditional transcriptomic techniques. It is a hybridization-based signal amplification system with a unique probe design that allows for simultaneous signal amplification as well as background suppression. The technology consists of three parts. The first part is a unique target probe that Advanced Cell Diagnostics designs against your sequence of interest. The second is a signal amplification system that generates a high signal-to-noise ratio. And lastly, you can visualize your data as a single RNA molecule punctate dot. This assay allows for spatial mapping of various types of RNAs, such as mRNAs, link RNAs, splice variants, highly homologous sequences, and point mutations in both cells and in intact tissues, all of which can either be visualized using a fluorescent or a chromogenic label. The assay can be performed on a wide variety of sample types, including FFPE tissues, frozen tissues. You can also run the assay on PBMCs as well as cultured cells. So how does the technology work? The two key features of the RNAscope technology are probe design and signal amplification. The oligonucleotide target-specific probes that you design are depicted as the Zs to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z complements and hybridizes to the target sequence, your sequence of interest, while the top of the Z is the base for an amplification structure. When the two Zs hybridize in tandem to the target sequence, it creates a binding site, upon which a preamplifier can bind and an amplification tree can build. Now, after these Z pairs hybridize with the target RNA, the preamplifier binds to the top of the Z, and then the preamplifier can bind multiple amplifiers, and further, each of these amplifiers can bind multiple labeled probes. These labeled probes contain either a chromogenic enzyme or a fluorophore that generates a signal detectable under a standard bright field microscope or a fluorescent microscope. This signal amplification strategy yields a high sensitivity and allows for visualization of target RNAs as a single dot. So now you may ask, how do we get that specificity? The specificity of the assay comes from these two different Zs. You need both of these Zs to bind in tandem for the amplification tree to be built and to see a signal. Just a single Z binding to a non-specific site will result in no signal. And so with the combination of this sensitivity and specificity, you end up getting that high signal to noise ratio, which makes this technology really, really good for different kinds of mRNA targets. We have a wide variety of product portfolios of both the RNAscope and the BaseScope assays. I won't go into too much detail as my colleague Andrea will talk a little bit more about it. But in a nutshell, we have both chromogenic and fluorescent assays that can either be performed manually or using our automated platforms such as the Leica platform. The two new assays that came out in the last 2 years was the HyPlex assay that allows for detection of up to 12 targets, as well as the microRNAscope assay. The BaseScope assay allows you to detect shorter targets. So it really covers a broad portfolio of different targets that you can visualize. So now let's go a little bit more over why is RNAscope ISH ideal for viral detection? What is the value prop of RNAscope for researchers that are studying viruses? So I try to break this down into different kind of categories, right? The characteristic feature of this technology enables one to overcome the various challenges that researchers face in detection of viruses. Let's start with the context. This in situ hybridization assay can be used with various sample types, such as the FFPE frozen and fixed frozen, as I mentioned before, that allows for detection of practically any target while providing you that morphological information along with that viral localization. This can help researchers identify entry portals, replication sites, disease sites, therefore further understanding the viral dynamics. Next, we go to sensitivity. As I mentioned before, the preamplifier and the amplifier tree system is built to enable for high sensitivity that allows you to detect low viremia. This helps in detecting viruses at very early stages of infection and latent stages that present lower transcripts and often present difficulties in detecting these viruses with some of the other traditional techniques. The specificity. As I also talked about before, the patented ZZ technology or the double Z technology provides high specificity of the assay. What this allows you to do is discern closely related viral species. Given the sequence is known, that if you know the sequence of the gene that you're looking for, ACD can design a probe for practically any species. This design process is rapid, which serves as a huge advantage for emerging viruses without available antibodies, for example, the COVID-19. The probes can be designed either single-stranded or can be made strand-specific to discern between various viral stages with hybridization to both the sense and the antisense strand. And this really allows you to look at both the replicating strand as well as the viral strand in the same tissue context, giving your data a lot of power. So RNAscope has an ability to detect a wide range of viral species. You can look at DNA viruses, RNA viruses, retrotranscribing viruses. The focus today, of course, is going to be this COVID-19 species. So now I'd like to go over some of the COVID-19 offerings. What are some of the products that ACD offers right now for enabling your COVID-19 research? This is a really good image that shows kind of like that broad offering range. You can first detect the virus itself by using our COVID-19 probe that targets the spike protein. You can also further your research by looking at the virus-receptor interaction by doing our duplex or our multiplex assays. In addition, as we all know, COVID-19 infection follows up with this immune response. And so you can also look at the virus receptor as well as the immune response that follows with this infection using our multiplex assays, or as they call the cytokine storm. And in addition, we've also seen a lot of researchers use RNAscope to look at various histopathological markers at the tissue of infection or the infection site. So it really allows you to look at this whole mechanistic action or the whole pathogenesis from how the virus infects the body to what receptors it interacts with and what is the body's response to that infection. So what is our coronavirus probe information? The standard coronavirus probe that ACD offers is this VNCOV 2019S probe. It is available and it targets that coronavirus outbreak that first happened in Wuhan, China in 2019. This probe was designed specifically to detect that spike S protein of this novel virus. Once again, going back to the specificity, it was designed such that it avoids cross-detection with some of the other SARS species, the SARS, the MERS, and other coronavirus family species. The probe can be used in a wide variety of animal models, such as the human, the mouse, rat, monkey, potential natural hosts, such as ferret, bat, etc. This probe can be used to study localization as well as infiltration of the virus in different tissues. Based on the data that has been published since the emergence of this COVID-19 virus, this given list of RNAscope probes are now made available, ready to use for customers to study the pathophysiology of the SARS-CoV-2 virus. I already talked a little bit about this before in the product offerings, but these are the actual targets that are already designed for you to order and use in your research studies. For viral detection, we have both the SARS-CoV-2 as well as the SARS-CoV-2 sense probe that allows you to now look at both the viral strand as well as the replicating strand. Again, the host cell markers, as a lot of you may know, some of the early host cell markers that were studied with SARS-CoV-2 were the ACE2, TMPRSS2, and cathepsin B and O. The cytokine storm markers or the inflammatory response markers, such as the IL markers, TNF alpha, and the MIP1 alpha. And the other thing I mentioned about is a lot of pathologists or scientists out there also want to look at the actual pathophysiology. And so they look at these histopathological markers like aquaporin 5, RAGE, and C16 in the lung tissue. In addition to studying these RNAscope markers alone, you can also combine in situ hybridization using RNAscope along with an antibody for IHC. This workflow has already been established. You can reach out to our support team and they can provide you more details on that. The way you would do this is you would perform the RNAscope assay first and follow it by performing your IHC or immunofluorescence staining. These are just some of the potential combinations or the different ways you can do this. You can perform the ISH assay for the viral probe, and then you can perform an antibody staining for the receptor probe, depending on what antibodies you already have available or you're using in your lab. So with all of these product offerings that ACD provides you and all of these various techniques that you can use, I'd like to now show you some of the examples of early papers as well as new papers of how researchers in the field are actually using the RNAscope assay to enable their COVID-19 research. So this first paper I want to talk to you about is basically the understanding of protective immunity to this SARS-CoV-2 is very critical for vaccine development and for public health strategies that are aimed to end this global pandemic. So a key unanswered question at that time was whether infection with this virus results in a protective immunity against re-exposure. Basically, if a person is infected, will this person develop a protective immunity? In this paper, scientists from Harvard Medical School, they developed a Rhesus macaque model for SARS-CoV-2 to study the pathological and immunological findings of COVID-19. They also used this model to study the natural immunity against SARS-CoV-2 after re-exposure. The RNAscope chromogenic red assay was used to visualize the SARS-CoV-2 viral RNA in the lung tissue, along with the detection of its replicating intermediate. So they basically wanted to look at both the replicating strand as well as the viral strand on the same tissue context. In addition to looking at the chromogenic assay, they also used the fluorescent assay to look at the SARS-CoV-2 strand. But in addition to that, they performed antibody scanning for both the virus as well as the nucleocapsid protein. The key message here is that RNAscope enabled the visualization of viral replication in the respiratory tract of these models that can be further used for testing vaccines as well as therapeutics, which is an emerging market in the field right now. This next paper came out of University of North Carolina from the Barrick Lab. The mode of acquisition and causes for the variable clinical spectrum of COVID-19 disease remains unknown. Scientists in this study, they used a reverse genetic system to study SARS-CoV-2 growth as well as pathogenesis. Now, they used RNAscope in two different sample types. They used it on cytospin samples as well as in tissue samples. The technology, using this RNAscope technology, they revealed that ACE2 expression in upper respiratory tract with increased sensitivity and efficacy compared to single-cell RNA sequencing. They used the RNAscope chromogenic red assay and established an ACE2 expression gradient in the respiratory tract and detected TMPRSS2 in normal human surface airway epithelium. To gain insight into pathways that might contribute to the dysregulation of ACE2 expression in cystic fibrosis lungs, they also tested the effects of selected cytokines on ACE2 expression in large airway epithelial cultures. Interleukin-1b, the dominant pro-mucin secretory cytokine in cystic fibrosis and non-cystic fibrosis secretion, upregulated ACE2 but did not upregulate TMPRSS2. This next paper came out of US Ambrid, and this was one of those early papers where scientists were trying to identify and evaluate commercially available reagents and assays for molecular detection of SARS-CoV-2 in infected FFPE cell pellets. Now, these researchers, they tested multiple IHC antibodies along with RNAscope ISH assay for viral detection in infected African green monkey kidney cells. In this first image, you'll see that they had used the RNAscope chromogenic assay to look at both the viral RNA as well as its replicating strand in FFPE cell pellets. But in addition, they also tested the RNAscope multiplex fluorescent assay to visualize viral RNA, again in both uninfected as well as infected cells. And they saw a strong signal of both the viral S protein and the replicating strand in the infected tissue, once again really emphasizing the sensitivity of this assay. In the same paper, lastly, they show the adoption of our dual ISH-IHC technique that I talked about earlier. In this paper, they combined both the RNAscope SARS-CoV-2 probe as well as its antibody in the same tissue section to simultaneously visualize both RNA and protein, truly highlighting the power of this dual ISH-IHC technique. This particular publication came out of Medical Center of Hamburg. Severe acute respiratory syndrome, or the SARS-CoV-2, preferentially infects cells in the respiratory tract, as we all know. But its direct affinity for other organs in the lung remains poorly understood. Researchers are still trying to understand how this infection affects other organs of the tissue body. In this study, researchers present data from an autopsy series of 27 patients that show that SARS-CoV-2 can be detected in multiple organs, some of them including the lungs, pharynx, heart, liver, brain, and kidneys. Now, they show a strong association between more than 2 coexisting conditions in patients and SARS-CoV-2 tropism in the kidneys. They detected the SARS-CoV-2 viral RNA with spatial resolution in tissues that were tested positive with PCR. They were FFPE lung and renal parenchyma samples, and they used the RNAscope red assay for this. Their results indicated that SARS-CoV-2 had an organotropism beyond the respiratory tract, including the kidneys, liver, heart, and brain. and that organotropism may influence the course of this COVID-19 disease and possibly aggravate some of the pre-existing conditions in the patients that were infected with this virus. There has been conflicting data about the possibility of vertical transmission of this SARS-CoV-2 virus from a mother to baby in the womb. In this particular study, Luisa Patani and other researchers studied 2 cases that were SARS-CoV-2 PCR positive mothers in the neonate and placenta. Now, they used the RNAscope Brown assay on the Leica platform to visualize the virus in the placental tissue, in addition to evaluating the tissue distribution. But they could do all of that while still retaining that morphological context. The RNAscope assay validated the presence of the SARS-CoV-2 RNA in the infected placentas, supporting the possibility of a vertical transmission from a SARS-CoV-2 from a mother to a baby in utero. Now, this image is from Arcana Labs and Yale School of Medicine, again related to that whole pregnancy outcomes, where researchers presented a case of COVID-19 positive patient who was in her 2nd trimester that was complicated by severe preeclampsia and placental abruption. Now, they wanted to distinguish between the physiological effects of preeclampsia and COVID-19 for better prognosis of pregnancy. They used the RNAscope Brown assay to visualize the SARS-CoV-2 RNA in the placental tissue. They also saw that the SARS-CoV-2 RNA staining pattern closely matched that of the ACE2 receptor pattern as seen on the Human Protein Atlas. This last paper was one of the early papers that came out of Chinese Academy of Medical Sciences. Plenty of diagnostic, preventive, and therapeutic knowledge on the virus was enriched from clinical studies since December of 2019. However, at that time, animal models, and particularly non-human primate models, were urgently needed for critical questions that could not be answered in clinical patients for evaluation of antiviral drugs and vaccines. So the researchers in this study, they evaluated various families of non-human primates to identify an accurate model to study the infection. They found that the M. mulatta was most susceptible to the infection compared to the M. fascicularis and the C. jacus. The RNAscope fluorescent assay was used to visualize the SARS-CoV-2 S protein RNA in infected non-human primate tissues to detect different degrees of viral distribution in different tissues. This particular paper is actually a really good example of a probe design. This paper came out very early this year when not a lot of antibodies were available. And the rapid probe design technology of RNAscope assay allows you to design for any particular species out there as long as the sequence is known. So with a lot of these publications out there, we have seen that the growth and adoption of the RNAscope technology is best exemplified by the number of our peer-reviewed publications. Our first publication came in 2011, and since then, there have been over 2,700 peer-reviewed papers using RNAscope technology in numerous journals, including many of the top-tier ones. It is highly relevant across multiple fields of research. Although this particular presentation concentrates on infectious diseases and COVID-19, you can also apply this technology in areas such as cancer, neuroscience, stem cells, development, inflammation, and many, many more. So with all of that, I hope I've convinced you today, and to summarize, that the RNAscope assay uses a unique probe design strategy to dramatically improve the signal-to-noise ratio. We currently have over 22,000 probes in catalog for various applications, but new probes can be designed and made within 2 weeks for any target and any species. The RNAscope provides both quantitative and spatial information on your targets in the complex tissue environment at a single cell resolution. The newest assay-based scope can detect splicing variants and short sequences down to 50 nucleotides, and that the RNAscope assay can be complemented with IHC or immunofluorescence for simultaneous RNA and protein detection on the same slide. With that, I come to the end of my part of the presentation. but I would like to give the cue to my coworker, Andrea, and he will go over the application of RNAscope on the Leica platform. Thank you so much, and if you have any questions, please ask them at the end of the presentation. Thank you.
Hi, everyone. Thank you for attending this webinar. I'm a field application specialist for ACD located in the Los Angeles area, and today I'm going to present you our RNAscope technology and how it can be used on the Leica BOND RX. I know Jyoti already gave you a more detailed introduction to the RNAscope technology, so I will briefly say only the following. RNAscope is an in situ hybridization technology that will allow you to detect and quantify a nucleic acid target inside a cell in a morphologically intact tissue using the microscope. It provides detection, quantification, and mostly morphological context of your targets. It consists of 3 parts. The first one is the RNAscope probe, which we provide and is designed for a specific target. We have thousands of probes available for many targets and many species. If we don't have one, we can make it as long as you can provide us with a target sequence to work with. The second part is the RNAscope assay, which includes all the amplifiers, preamplifiers, and reagents necessary in general for building the signal on top of the probe so that it can be visualized. Finally, the third part is the imaging of your sample under the microscope and the visualization of the RNAscope signal, which consists of dots, where each dot represents a target. The location and number of dots tell you about how much and where your target is expressed. An important thing to know about this workflow in general is that when done manually at the bench, it takes approximately 1 to 2 days, depending on the type of assay. A single-plex assay may take a day, a fluorescent assay, 4-plex may take up to 2 days. So basically, having access to the Leica BOND RX or RXM is a great asset, because the major feature of this technology is that it can run up to 30 slides completely unattended. The run is approximately 10 hours, 11 hours. We generally run it overnight. And it's designed to process mostly samples prepared as FFP samples, formalin fixed, paraffin embedded, but it can also process fixed and fresh frozen samples. It just takes a little bit more planning. Before I get into the details of the RNAscope on the Leica BOND, I want to give you a quick overview of the portfolio of the RNAscope assays that we provide that can be used on the Leica BOND. So the RNAscope assays that we provide that are available are the single-plex brown and single-plex red RNAscope assays. The single-plex brown uses HRP as an enzyme and DAB as a substrate to give you a brown pigment. The RNAscope red is giving you instead a red dot, a red pigment, and it's using alkaline phosphatase as an enzyme and fast red as a substrate. They're both single-plexes, so they both detect only one target, and they're perfectly compatible with the Leica BOND RX system. The other type of assay that we offer is the RNAscope duplex. The duplex assay will allow you to detect 2 targets at the same time in the slide. One is going to be one color and one is going to be another color. So you're going to need 2 probes, RNAscope probes, respectively known with a different channel. That's a topic that will be explained better by technical support if you need to. And if you wish to run this assay entirely on the instrument, you can use only the red and brown pigments, but you have the options to do the red pigment on the instrument, and if you prefer to have the second signal as a green dot, that can be done offline. It's a very fast procedure done at the bench immediately after the run. So brown and red online on the instrument, and red and green offline only for the green part. The other RNAscope assay that we offer that is used a lot with the Leica system is the multiplex fluorescent assay, RNAscope assay. It will allow you to detect up to 4 targets at the same time, but can also, of course, detect less targets. You will need to get fluorescent dyes from, known as also TSA plus dyes or opal dyes from Leica Biosystems. And you will need to have up to 2 to 4 RNAscope probes. They all have to be different channels, so there is no crosstalk. And this procedure, again, is perfectly compatible with the Leica BOND RX. Then the other 2 assays that we offer are the BaseScope assay and the microRNAscope assay. The BaseScope assay is a singleplex type of assay for the detection of very short sequences down to 50 base pairs. So if you have a very small target because the region nearby is not known or is too conserved in the genome, or if you have an exon-exon junction or something just simply very small to detect, and you cannot do it with a conventional RNAscope assay because that one covers a 1 kb region, you can use the BaseScope assay. That's because it covers only 50 base pairs. This is offered for the Leica BOND as a singleplex red option and assay. And the newly born in the ACD company product is a microRNAscope assay that will allow you to detect microRNAs using the red chemistry, just like the singleplex red. So, regarding the detection of COVID the virus that causes COVID, as you well know, COVID involves wide variety. It's quite a complex mechanism that involves having the virus attacking the host cell, and when that happens, we have the viral genome, and then we have the production of a replicating strand inside the host cell. And the downstream cascade of events is basically a big inflammatory response, and of course the disease that we all know as the COVID-19 disease. This mechanism involves a wide variety of targets, and we do offer them as part of our portfolio as an RNAscope product. So we have several different probes that will allow you to detect the virus strand, the positive strand of the virus. We have probes that will allow you to detect the replicating strand inside the cell. We have 2 types of probes for the replicating strand, one that is exactly complementary to the positive strand, and one that is instead not complementary. It's just a little bit downstream the region that will allow you to do duplexing. And then we have probes, of course, that are available for the detection of the markers and the targets that usually are involved in this mechanism, like the ACE2, cathepsins, and so on, and of course all the inflammatory genes. These probes are available. You can go to our website. If you go on the top right side, you can click on search, and basically you can type the name of the target that you wish to detect. And the example that you see here on this slide is for one of the probes. It's for the replicating strand. And when you go to each probe specification page, you can see exactly what is detecting, where this probe has been designed, the accession number, the target region, as you can see here, that's exactly where the Z probes are binding, and the length of this probe, in this case, is the 40 base pair region. So you get to see for each probe involved in the COVID-19 infection mechanism, the specification of the design of the probe. Now, these probes are available for the RNAscope as an RNAscope assay on the Leica system. ACD offers 2 product configurations for running RNAscope on the Leica system. One is the LS reagent, and the other one is the LSx reagent. The question is, how do they differ? I'll start with the simplest one. The LSx product configuration is available only for the singleplex brown RNAscope and singleplex red. This configuration, the LSx, is the most user-friendly. The user does not need to transfer any reagent from the ACD vials or bottles into the Leica containers. Everything is already prepackaged at the factory in the Leica containers. The reagents are already loaded onto the software. We'll see that a little bit later. And all the user needs to do is to simply slide these trays, the reagent trays, into the instrument, load the instrument with the slides, and of course, the user needs to scan these barcodes that you see on these trays, but that's a very simple action. You scan the barcode and you click and you say OK on the screen. And that's that's it. You basically just run the instrument, and for 10 hours, you're going to have your instrument running from beginning to the end. This is the simplest and fastest and most convenient type of configuration, the LSx, which, as I said earlier, is available only for red and brown singleplex chemistries. We do have the LS product configuration, which instead is available for more RNAscope assays. It is available for the brown and red singleplex, just like the LSx, but it's also available for the duplex RNAscope assays, the fluorescent multiplex assays, the BaseScope, and the microRNAscope assays. So there is a wider portfolio. There are more options. The difference, though, with respect to the LSx, is that it's not as user-friendly. That being said, it's very easy to set it up. The reagents are coming from our company in a box, and they are in bottles. And all you need to do, what the user needs to do, is to transfer these reagents in the appropriate containers. The containers must be obtained from Leica, and just like other reagents for running the instrument must be obtained for Leica. So this is the running of an LS RNAscope experiment is done with reagents that are coming from both companies, of course. So the only convenience is basically the loading of the containers, which is not prepackaged. And one important thing to know is, which is very nice, is that none of these reagents need to be warmed up before loading. The running of the experiment on the instrument, of course, being an instrument, is connected to a computer and is driven by a software. I just want to spend a little bit of time on the software and tell you that currently the Leica software is the 6.0 version. There are other versions out there, mostly the 5.2 and the 4.0. I recommend you to update if possible, because the 6.0 is the version that offers it's extremely user-friendly. You get to see here a quick screenshot on the far right of the slide. It's very user-friendly because it will allow you to create and customize the protocol in a very easy way. You can choose between open and closed washes. You can choose the temperature of incubation, the time, and what items need to be added to your protocol. You can do these things also in the older versions. It's just not as user-friendly. All RNAscope assays can run on the version 6.0, so no worries about what you can run on this version. The only one you need to be aware is the newest microRNAscope assays that can run only on the 6.0 and cannot be run on the older version. So if you have the 6.0, you're basically perfectly safe and you can go ahead. There is another important thing in addition to the software version that a user needs to be aware of, and that would be the data version, what we call more simply as BDZ version. LS RNAscope assays require BDZ 11 type of data version. If you wish to run the very user-friendly LSx red and brown chemistry, you need the BDZ version 15 or above. And if you're running our most recent microRNAscope assays, you need the BDZ 20 or above. So when you go to your software, please go to the section where you can look what type and version of software you have, like you see here on the screen, and be sure that look at the data version number and be sure that it's compatible with the RNAscope assay that you want to run. All right, let's look very quickly at the workflow and how it works. This is not meant to be a training. We do this on site in your lab with one of us. We come to your lab, in other words, but I'm going to give you anyway a quick overview. The running an RNAscope assay basically involves the following steps. The first step is to add reagents to the software. The software comes with a series of reagents already pre-programmed at the factory. All these reagents have an asterisk next to them, so you know what's hardwired at the factory. Some of these reagents can be used for RNAscope, some others are not usable, or better, are not there to run RNAscope. The user can easily generate them. So you need to do what we call user-made reagents. All these reagents will not have an asterisk next to them, and you can give them a name. Just be aware you cannot rename a reagent twice with the same name. You need to change the name a little bit. And just as a reminder, if you're working with the LSx reagents, you do not need to do any of these. But anyway, you will need to add and generate some reagents pertaining to the RNAscope assays that you want to run. The next step will be to set up the protocol. So the protocol is basically a set of instructions that the machine needs to have in order to execute a specific workflow. In this protocol, you have the reagents you generated, and you have also the timings and the temperatures at which these steps need to be executed. Protocols, just like reagents, some of them are pre-made at the factory. All of the pre-made ones come with an asterisk next to it. If you need to make a protocol, and you will need to make a protocol, those will not have an asterisk next to it, and you can name them whatever you want, you just cannot use the name twice. Again, if you're running the LSx chemistry, you do not need to worry about generating a protocol. And just to give you a little bit more of a detailed explanation on what it means to make a protocol, basically what you need to do is open a pre-made, preset protocol that is already on the Leica system, like in this case, the ACD 2.5 DAB revision B protocol, which you can see here, and modify it according to the user manual that you have in front of you when corresponding to a specific RNAscope assay. The modifications that you need to do are basically in front of your eyes. You need to be sure that all those steps that you see on the far right of the slide are there, that the reagent that is listed is exactly that one, and that the incubation times, temperature times, and washes, whether they're open or closed and so on, are exactly as per outlined in the protocol table. Once you're done making the modifications, then you can save the protocol and you can use it in the future multiple times. So this is mostly a one-time thing that you need to do. And let me remind you that we do this for you. The field application specialist from ACD will come to your lab and generate the protocol for you. And we do as many protocols as you think you might need. If down the road you need more, you can try to do it yourself, or we can come back and do it for you. But basically, making a protocol involves taking an existing protocol, edit it, modify it so that it can run properly the RNAscope assays. Once the protocol is made, the next step is to basically register the reagents, and that means you're scanning the containers with the reagent so that the instrument can link a certain container to a certain reagent present on the protocol. That's very important, and is done with a scanner. And once you're done, you have done this, you basically you can start setting up your experiment. You're going to click on Slide Setup tab, like it's shown here on the slide. Subsequently, you're going to give a name to your project or study. Here is where you're going to decide whether you need the bake and dewax steps. Those are necessary when you're using formalin-fixed paraffin-embedded slides. If you don't have those kind of slides, you can omit it. And Then you're going to click on OK, and then you're going to add for each slide a name and a probe name. So you're basically giving to the software information about every slide. And every slide is characterized by a sample name and a probe name for sure. Then, of course, to that slide, a certain protocol is applied, a protocol that you made, including all the pretreatment conditions, which are the two things you are going to mostly work with and modify in order to optimize your assay. Once you're done adding the slides get added in the computer, in the software, and finally you are ready to print out the labels. The labels will go on the slides, on the glass slides, and then you're going to load the instrument. This is a quick overview of the instrument. It's the Leica BOND. You have bulk reagents at the bottom. Just be sure the reagents, there is enough to run the number of slides that you want to run. If you don't have enough, the instrument will tell you, the software will complain and tell you. Then you're going to load the trays into the instrument, and the trays that you can load are 3. There are 3 slots. You can load up to 3 different trays. Each tray can host 10 slides. And on the far right of the instrument, you are going to load the trays with the reagents. And that's where all the reagents are. And at that point, you're ready to run. And at the end of the day, as I said, it's an overnight run, you're going to have your slide completely stained. All you need to do is just cover sleeve them. Regarding sample preparation for the COVID detection, it doesn't really matter much whether you're detecting a target related to the COVID disease or not. This is the typical sample preparation that we recommend for any tissue that is formally fixed, paraffin embedded, so we recommend to have it fixed for between 16 and 32 hours. We recommend to cut it at a thickness approximately of 5 microns. We recommend you to use and put your tissues on positively charged slides, and once you're done, you can store your slides in a desiccant at room temperature and use them, please, within 3 months. All right, in regard to the pretreatment conditions, those are basically very important because it's where you're going to do the majority of your optimization. So the workflow runs a series of steps that are pretty much set in stone, but before that, you need to decide how long and how warm is your target retrieval step will be, and what type of protease you will use and for how long and at what temperature. That's where you're going to do your optimization. So in the protocol, in the software, you can choose target retrieval, known as epitope retrieval, which on the software appears with the letters H-I-E-R here. And there you can choose between 88 degrees or 95 degrees and how many minutes. For the protease detection, usually we don't really play much with that, but in case you need to, especially if you're using fixed frozen or fresh frozen tissue, then that would be the option usually are to use either the protease 3 or the protease 4. So the protease 4, which is a stronger protease, it's an option. And finally, to give you a quick summary, I can say this: the RNAscope assays are available to run on the Leica system. You can run up to 30 slides efficiently and completely unattended. Brown and red chemistry singleplex are available in LSx format, extremely user-friendly, or I'd call it plug-and-play workflow. Brown-red duplex, multiplex fluorescent, base scope, and microRNA assays are available in LS format, which requires reagents filling into the containers, protocol editing. I recommend you to please fill the containers in the proper way as per manual. We have the option of 30 ml containers, 7 ml containers, 6 ml containers, and especially for the 6 ml containers, if you're just loading just what you need for that specific run, do not forget to factor the dead volume, which would be 600 microliters. So you need to put in that 6 ml container, whatever you need, plus a 600 microliter. An important thing to know about the Leica system is that you can delay the start of a run, so you can have it finish at whatever time and day you want. We do recommend you, however, that when it's finished, to basically take the slides out and cover slip them as soon as possible, because you don't want to lose your counter stain. And this is pretty much it. Thank you very much for attending the webinar. I'm now going to hand it to Damian Cockfield, and have a nice day.
Hello, my name is Damian Cockfield. I'm the BOND product manager out of Leica Biosystems, Melbourne. I'm responsible for the BOND research and BOND clinical systems. And I've also, in my previous roles in Leica, acted as a laboratory scientist as well as project manager, and actually worked directly with ACD on the RNAscope technology. My talk is a bit of an extrapolation of where the first two talks finished. I'll talk about the RNAscope workflow on BOND-RX and the benefits of full automation to your research. The BOND-RX research data from Leica Biosystems helps researchers drive their biomarker application discoveries forward by providing the quality and flexibility whilst enabling the automation of IHC and ISH emerging tests. Whilst the talks from Jyoti and Andrea will more focus on the explore your ideas phase, this talk will focus on how automation can accelerate your research program through the benefits of automation. But first, let's take a step back to why we're talking about RNA detection in tissue. DNA and genes code for everything that our body develops, good or bad. DNA is the blueprint for life, and that blueprint is for making proteins. So through a process of gene expression, the genetic information is transcribed from DNA to RNA and then translated to messenger RNA. It is this mRNA that instructs the synthesis of protein. Protein, being the normal target for IHC, is where the bulk of the work in research and diagnostics on tissue has been done until recently. So why RNA and why detect in situ? Using RNA probes, there are over 30,000 markers available with the ACD RNA-ISH technology, opening up a plethora of opportunities to discover new information about what is occurring within tissue. RNA markers can fulfill a need where antibodies may be ineffective or not available, chimeric proteins, for an example, or more importantly with today, viral markers like COVID-19 being of real interest. And this follows on the back of market acceptance of using RNAscope for other viral targets like HPV probes. So why we want to detect RNA in situ is pretty clear. Cellular context is vital to a biomarker's clinical relevance, and mRNA gives clues as to what's happening in the tissue at the time of excision. But why RNA over DNA or protein applications is not so obvious, and largely that's because RNA-ISH has been an inferior test for many years. But that's no longer the case, and RNA-ISH bridges the gap between DNA and protein in the cell and allows researchers to see the current state of what is happening in tissue. Why RNAscope as a solution was made pretty clear in the previous two talks. At the end of the day, it comes down to an increased confidence in your results, namely around specificity, sensitivity, and the ability to visualize the resulting outputs. Due to the proprietary technology, only the target signal is amplified. This, combined with background suppression, ensures target-specific binding. The sensitivity of the assay allows for detection of single RNA molecules in single cells as punctate dots in their morphological context, which can be easily visualized with standard microscopes and then quantified manually, or digitally scanned on products like the Aperio GT450 and quantified with the Aperio RNA-ISH image analysis algorithms. RNAscope as an in situ hybridization technology based on unique probe design and RNAscope is an in situ hybridization technology based on a unique probe design and signal amplification method. The technology has the sensitivity to detect most genes in situ, as well as simultaneously quantify multiple RNA transcripts. But as in situ assay, RNAscope assay involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to the RNA target of interest. The specific signal is amplified using the ZZ probe system, which is subsequently detected and quantified. The assay is extremely universal as well as rapid and scalable. Overall, the technology is broadly applicable for the detection of any expressed biomarker in tissue. Here is the workflow for the RNAscope assay. Slides are baked and dewaxed, then pre-treatments are applied following the RNAscope probe, hybridization, multiple rounds of amplification, chromogenic conversion, and counterstain. Manually, this process is quite long and has to be done over 2 days, separated across days with pre-staining and post-staining. This workflow doesn't show the additional washes and labour time required, but those of you experienced in IHC and ISH staining will note that this test will benefit heavily from automation, as you can load your test slides onto BondRx at the end of the day and let it run everything overnight and take off the complete sample in the morning. As mentioned in the previous talks, the RNAscope product range comes in a range of formats: manual, semi-automated, and fully automated. Automation with the BondRx provides you with the ability to run slides overnight, so you're not wasting your day on manual applications. It provides the ability to run up to 30 slides of the same test type or run parallel multiple applications like DNA, RNA, and IHC all together. And finally, the BondRx and the unique cover tile system protects your samples and allows for consistent processing time after time. And here are the 2 fully automated products on BondRx, the RNAscope 2.5 LSx brown and red kits. As you can see, the reagents within the kit are all barcoded, so you just have to scan each of the barcodes and BondRx will know exactly what the kit is. That's it. No decanting, no prep and reagents, just scan and load. So first off, we have the RNAscope 2.5 LSx brown kit. So the kits are all RUO labeled or research use only. They all come in that ready to use format. They are arranged in a 2-tray system with detection and amplification reagents. Each of the kits are 60 slides and account for 15 different staining runs on the research system. The brown kit uses the same DAB and hematoxin that is also used in the BOND Refine kit. So those of you who already use BOND will see that it has that similar in look and feel to make it easier for you to assess. The red kit has a similar format with the 60 slides, requiring batching of 4 slides or more. And really, with lung being an area of interest for COVID-19, red detection may be more appropriate detection system for some of those research applications, as it provides a color contrast between the sort of browny black pigment that you sometimes see in lung tissue. So both of these applications can be ran overnight. The key with automation isn't just about automating the steps of staining, but it's also making the interactions with the system simple and enjoyable for you, the user. That simplicity, though, can't be at the cost of flexibility, especially for researchers who require the freedom to optimize. RNAscope 2.5 LSx comes with pre-configured protocols to allow you to plug and play your reagents and test so that you can be up and running straight away with the option of preloaded heat retrieval protocols, as well as enzyme retrieval protocols to provide retrieval flexibility based on your tissue samples. Added to that, the open protocol system allows you the flexibility to adjust your protocols as needed. But having used the products myself, I know they work pretty well out of the box. And in summation, I hope you've been able to get a glimpse of how RNAscope and BondRx work together to provide a path for your research. Broad adoption of new technologies and targets comes when the barrier to adoption is low or the return on effort is high. Automation of RNAscope on BondRx and the great range of compatible probes like the COVID-19 probes provided by ACD helps take a niche manual test utilized by highly skilled and potentially time-poor researchers to allow simple, easy-to-use solution that you can trust so you can focus on the results and not the application. Thank you.
Thank you, Jyoti, Andrea, and Damian, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. I think we have time for about one or two. So, Andrea, can you do IHC or IF on the Leica together with RNAscope?
Yes, hi. The answer is yes, you can. We generally do RNAscope first, and then we do immunofluorescence or immunohistochemistry. And yes, you can do both on automated fashion on the Leica BOND.
Wonderful. And Jyoti, this one's for you. Why is the scientist research on Rhesus macaques instead of ferrets for COVID-19?
Hi, Sabrina. So I believe the question was more related to the paper that was discussed. That paper had come out early during the COVID-19 outbreak in 2019, where a lot of other animal models were not studied. That's why the focus was more on Rhesus macaque models. But we have seen an uptake of publications in a lot of other models due to our rapid probe design ability to design the probe for any kinds of species, if that answers the question.
Yes, thank you so much. And thank you again, Jyoti, Andrea, and Damian. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank our speakers for their time today and their important research. We would also like to thank LabRoots and our sponsors, Leica Biosystems and Bio-Techne, for underwriting today's educational webcast. You can view this webinar on demand. LabRoots will alert you via email when it's available for replay. That is all for now. Until next time, goodbye.